I really look forward to introducing you to Dr. Tim Oldham in a minute. As you would know, we've got 5 ASX listed companies today speaking on the topic of addressing the need for change. Dr. Tim Oldham is CEO of AdAlta, and in case you're not across AdAlta, it's an innovative clinical stage biotech company developing a unique range of new drug treatments. It's now utilising the power of its Ibody technology to create a pipeline of new drugs. And an Ibody is a unique human protein that combines the advantages of small molecules and antibodies in one powerful treatment. So Tim will explain how AdAlta has grown from being a single product company not all that long back to now really utilising that Ibody platform to respond to emerging opportunities and needs. The company's got programs in fibrosis, in PET imaging for immunotherapy, and as of recently, precision engineered and targeted CAR T cells for solid tumours, so covering a lot of ground. So with that, Tim, I'd like to ask you to now start screen sharing, and I'll hand over to you. Thank you.
Thank you very much, Jane. Thank you everybody for joining us today. I'm going to take a little bit of a different take to the approach or the description of AdAlta to the usual one for those who've heard it before, and for those joining us today for the first time to hear about AdAlta. Really want to focus on what are the needs of the industry and frankly our patients ultimately that we're trying to solve. So AdAlta is an ASX listed drug discovery and development company. On the next slide, because we are listed, we'll be making forward statements today, and they are subject to the usual disclaimers. Our purpose is to use our unique Ibody technology to create multiple novel therapeutics for diseases where traditional antibody technologies have struggled. The next slide really summarizes where AdAlta sits today. We have an Ibody platform, which I'll talk more about in a moment, which is a powerful drug discovery tool for addressing targets that traditional antibodies struggle to serve. We have a lead asset that's already in the clinic, and we're preparing an inhaled version of AD214 for phase 2 studies in Idiopathic Pulmonary Fibrosis. We have 2 existing co-development collaborations with GE Healthcare and Carina Biotech, and we're building out a pipeline of additional assets over time to really leverage the full value and realise the full value of the Ibody platform. Today, I really want to focus what we do around the unmet needs in our industry and for our patients today. And so I'm going to cover 4 areas that are summarised on the next page. The first one is that antibody drugs, while revolutionising the pharmaceutical industry about 30 years ago, still can't do everything. And so we do need new drug discovery platforms to address some of the targets where antibodies struggle. And this is where the Ibody platform comes into play. Secondly, I want to talk about idiopathic pulmonary fibrosis. This is a degenerative and fatal disease where our lead product is offering new hope and a new avenue of dealing with this. The third need is around immuno-oncology drugs, which are revolutionising cancer treatment for some people, but we can't work out who they are. And so the need I'm going to talk about today is how do we identify the responders early? And finally, a subset of immuno-oncology is the new therapeutic area of CAR T cells. This is a cell-based living therapy, and this has provided fantastic new hope for blood cancer patients. But the challenge we now face that AdAlta is seeking to address is how do we take that same hope to patients with solid tumours? So let's turn to the first of these topics. Antibody drugs can't do everything. The next slide will show how Ibodies are different to traditional antibodies. Traditional antibodies are represented by the large picture at the top, in the middle of the top row. They're very, very large molecules. They confer fantastic specificity, ability to bind to a specific target compared to a small molecule, but it's that sheer size that makes it difficult to generate and penetrate some targets. Ibodies, shown on the top right, are a very small format antibody-like molecule. They are designed to mimic the properties of the immune system found in sharks. This small format antibody has the ability to penetrate some of these difficult targets, And importantly, because of its small size, as shown on the bottom, we can also use it in a modular format to create a whole range of diverse applications. And so we think about this Ibody platform as illustrated on the next page as a very powerful drug discovery platform. It can address a wide range of important target classes, and we've hit more than 25 different targets with the Ibody platform to date. An important class of these is a class of molecules or receptors called G protein coupled receptors. These represent about 30% of all marketed drugs today target a G protein coupled receptor, but only 2 antibody drugs have been successful doing that. And so we're missing that specificity for this whole important target class. I talked about different formats for Ibodies, and we can combine them in multiple different ways. We can use the Ibodies as a direct therapeutic, as shown on the right on the bottom. And this is where AD214 comes in, where binding the target is what generates the therapeutic effect. But we can also use the Ibodies as a targeting agent to carry a therapeutic cargo or payload, if you like, more precisely. And this is where our Granzyme B or GZMB program with GE Healthcare comes into play. Targeting a therapeutic moiety is also what's behind our CAR-T initiatives. But that combines an additional component, which is the ability to combine Ibodies together to create bispecifics or multispecifics. And this creates even more enhanced targeting capability. And we're aiming to combine both of those with our CAR-T platform. So Ibodies really are this powerful drug discovery platform that can help address some of the challenges that our industry still faces in how do we get to some particular target classes and some particular modalities that have proved challenging. So let's now turn to the first application of Ibodies, idiopathic pulmonary fibrosis. The next slide summarizes this disease. It's a degenerative, progressive, irreversible lung disease where essentially the lungs are scarred, progressively lose their function. Median survival post-diagnosis today is still less than 4 years. There are 2 marketed drugs that generate $3 billion in sales per year, but they still do not work effectively. They have nasty side effects and they slow progression, but do not halt progression and certainly don't reverse disease. So there's a crying need for new therapies, and unfortunately, the burden of fibrotic lung diseases following COVID-19 infection is also likely to be high. And so there is an even greater need and urgency for us to find alternative solutions to this particular challenge. So AdAlta is deploying its Ibody technology and its unique ability to hit these difficult drug classes, including G protein coupled receptors, to find a new pathway to treat IPF. And on the next slide, we illustrate what we're trying to achieve. There's lots of text, but focus just on the images. The top row shows normal human tissue on the left and fibrotic tissue on the right. And you can see the destruction of the normal tissue architecture. You can also see brown staining. And this is a marker of CXCR4. This is the receptor that we're targeting with AD214. It's a G protein coupled receptor, one of those that's been very, very difficult for antibodies to address. And it's heavily implicated in the development of fibrosis. Now, on the bottom panel, you can see the impact we're hoping to have. This shows a mouse model of lung fibrosis, and the purple staining here is now collagen, which is a measure of scarring. It's what causes stiffness when you cut yourself. It's the same thing that causes the lungs to stiffen with IPF and therefore reduce your respiratory capacity. The left panel shows the normal lung tissue. The middle panel shows diseased or fibrotic lung tissue, lots of purple staining and that classic destruction of the tissue architecture. The right panel shows what happens when we treat with AD214. And you can see that we have much lower collagen production and a much more normal tissue architecture. So this is the outcome we're trying to achieve with our lead candidate, AD214. We've just completed our first phase 1 clinical trial with AD214, and the results are summarized on the next page. Detail is here for our presentation, which can be found online, but the key messages are AD214 in single and multiple doses in healthy volunteers was very well tolerated, and we did not see dose-limiting toxicities or adverse events of clinical concern. Importantly, we're able to show that AD214 engages the CXCR4 receptor using markers in the circulating bloodstream. And we also found that and understood the pharmacokinetic and tissue distribution profile. And while the pharmacokinetic profile was satisfactory, parallel preclinical studies also showed through PET imaging that we were getting a significant amount of the drug was distributing to the liver of mice and non-human primates, as well as binding to CXCR4 where it was found. And so this has caused us and enabled us actually to pivot very quickly. We've talked about addressing changing needs. This is where I'm really proud of our operational team for pivoting very, very quickly to an enhanced and superior route of administration for AD214, which is inhalation. And on the next slide, we talk about why we did this. Firstly, we've always believed that a more convenient route than intravenous administration was important for AD214, and that eventually we would move to something like inhalation. The rapid liver distribution we've seen in preclinical studies using PET imaging shows that the bioavailability of the drug by intravenous administration is lower than we would have expected. Nonetheless, we are still capable of binding CXCR4, and we still see efficacy in the animal models we've studied to date that show this localization. We also see that liver distribution does not appear to affect the safety profile of the drug. So all of that's pointed us to direct lung delivery of AD214 to achieve a therapeutic dose at a much lower level and a much more convenient cost of goods than intravenous delivery. And we now know that we can deliver that outcome within the time frame of the commencement of our next clinical study. So on the next slide, reiterating the benefits of our phase 2 program planned with an inhaled formulation, we expect to generate improved bioavailability. This will be a way more convenient formulation for patients. It will be a much lower cost of goods, and it will create diversified partnering options for us in the long run because with indication-specific formulations, we can now partner the drug by indication. So not only are we adapting to a crying need for improved therapies for this particular indication, we're also adapting our research programs in real time as the data tells us and drives us to achieve a better product for those patients. So moving on to our second opportunity, the field of immuno-oncology drugs. And on the next slide, we talk about the opportunity that immuno-oncology drugs have created for cancer patients. And essentially these drugs reactivate the patient's own immune system to fight cancer. The problem is that while this is going to be a $95+ billion market, only 20 to 40% of patients respond to these immuno-oncology drugs. And today we can't tell who they are until their tumor progresses, which is too late. We know that a molecule called granzyme B is produced by immune cells to kill cancer, and this represents a potential biomarker of these immuno-oncology drugs to tell us who the responders are. On the other hand, PET imaging is being used regularly within cancer to improve diagnosis. And so the combination of being able to PET image granzyme B could enable us to identify the responders to immuno-oncology drugs much earlier. And the PET imaging market is a $6.4 billion market today, with the largest products generating $400 million in sales. And importantly, PET imaging agents have a much shorter development time than therapeutics. So when GE Healthcare came to us and said, can you help us develop a PET imaging agent against granzyme B, this was a really exciting opportunity for us. And our collaboration is summarized on the next slide. We bring our Ibody technology to find Ibodies that bind to granzyme B and carry GE's PET imaging traces, fluorine 18 in their case, to granzyme B. GE are responsible for the preclinical and clinical proof of concept and commercialization, and they paid us upfront milestones and research fees for doing the Ibody discovery. And so far, we've earned more than $1.5 million of revenue from this collaboration. This is now in preclinical proof of concept, and we hope to be able to provide some further update on that progress early in 2022. So if I turn now to the final opportunity that AdAlta is addressing today, and this is the field of CAR T therapy. The next slide explains what CAR T therapy is. Essentially, we are taking a patient's own immune cells, from which, remember, the cancer has been hiding. And we reprogram them in the lab through genetic modification to insert a receptor called a chimeric antigen receptor, or CAR, so that the immune cells can now see the cancer. These are expanded in the lab and given back to the patient. And now the patient's own immune system is actually not just primed, but actually engineered to be able to fight the cancer. The first ever CAR T products were approved for blood cancers in 2017. They already generate $1 billion in sales a year. We expect the market to reach over 20 billion by 2028. And a big part of that will be driving from blood cancers into solid tumours. And these will represent more than 50% of the market by 2030. We are collaborating with Carina Biotech, as outlined on the next page, to generate precision-engineered Ibody-enabled CAR T therapies to provide new hope to these patients with solid tumour cancers. We're combining our Ibody capability to target tumours with Carina's world-leading CAR T delivery technology to solve other problems. And the next slide really summarises the three big problems of translating this wonderful innovation for solid tumours, for blood cancers, into solid tumours. The first one is we need to improve precision. Blood cancers are relatively easy to target, but there are not very many tumour-specific antigens. These are what the CARs actually bind to, known for solid tumours today. And this means that we can either damage healthy tissue, or that the tumour, because it expresses many, many different antigens, can actually escape. AdAlta's Ibodies are specifically designed to enable us to access different and new difficult antigens. And the small size makes it much easier to create bispecific or dual CARs that target multiple antigens. And this could enable us to improve specificity and reduce the risk of tumour escape. So from a precision point of view, AdAlta's technology is ideally suited to addressing the next generation of solid tumour CAR Ts. But we need to be able to have the CARs perform. They need to be able to get to and into the tumour. And we need to be able to have them persist in an environment within a tumour that is designed to suppress the immune system. And this is where Carina's CAR T manufacturing and chemokine receptor platform comes to the fore. They have a best practice manufacturing process that produces a more robust, younger T cell as a result of the manufacturing process. And their receptor platform enables us to engineer in additional features that improve homing or performance, as well as improving resilience to the tumour suppressing microenvironment. So it's an ideal combination to really take the transformational outcomes that these CAR T cells are created for blood cancers into solid tumours. So where does that leave AdAlta as a whole? On the next slide, you can see really what our business model is and where our pipeline sits today. Our platform enables us to create multiple assets. We're generating a pipeline of wholly owned internal assets. 8214 is the lead candidate there, already in the clinic, and we're going to add another couple of targets within 2021. And then we enter these co-development collaborations to combine our technology with other companies' technologies to solve other unmet needs, and GE and Carina are the first two of those that we have. This puts us, as you can see from the next slide, really at the beginning of a journey that several other companies have undertaken before us. And this is leveraging a platform with unique abilities to hit difficult targets and turning that into a multi-product, multi-partner opportunity. Ablinks is the company that's most similar to us, had a very similar platform to the Ibody, and from where they got their first program into the clinic in 2007, they grew to a $5 billion company when they were acquired by Sanofi 10 years later. They're not the only one. You can see two other examples on the right-hand side of this slide. I'm very proud of the team that we have working together to drive this technology forward. You can see them on the next page. With their backgrounds, and you can see that our team is deeply industry experienced across all aspects of drug development and commercialization for biologics and for respiratory diseases in particular. Moving on to the next slide, just summarising who we are and where we're up to today. Our Ibody platform is the engine room that enables us to create value over time for our shareholders. Our lead asset is advancing into phase 2 clinical trials. Its initial market opportunity is a $3 billion market. And we already have 2 co-development collaborations with GE Healthcare addressing a $6 billion market and with Karuna Biotech addressing a $20 billion market. I hope we've been able to describe not just how we're addressing unmet needs and adapting to the challenges of advancing therapies and technologies, but also that we have a very clear picture of how we're going to build value in the company over time. Our team is fantastic. I'm very proud to be leading them. And we haven't talked about our news flow. It is available on our website, but we do have regular milestones coming quarter on quarter to demonstrate de-risking of AD214 in particular and the expansion of our other programs. And so with that, I will stop and answer any questions that anyone may have.
Thank you, Tim. I might ask you to, oh, you've done that. Great. Thank you. Okay. So we do have a number of questions. I'm going to kick off with my favorite one though, which is, of course, today, nanobodies hit the news. I don't know if you've seen that. Yes. Of course, a coronavirus treatment made from llama antibodies was shown to be one of the most effective treatments to date. So an astute audience member has asked, with Fifi the camel news overnight, could you please compare and contrast shark antibodies with camel antibodies?
Wow, that's a really great question. So at a high level, sharks and camels antibody systems are very, very similar. Independently, they evolved these small format, what we call single domain antibodies. They're both around about the same size, and they're about a 10% the size of a traditional human monoclonal antibody, and that small size is what confers their advantages. There are differences, and the differences go down to the biochemistry of how the proteins are actually put together. The camelid system looks a little bit more like the binding domains of a human antibody. So we think that the sharks actually have a degree of flexibility, again, on top of what the camelid antibodies have. But pragmatically, that whole class of single domain antibodies there's a relatively small number of companies working on these, a lot of academics working on them, and that's because they have significant power to address challenging targets. And yeah, we've been monitoring the COVID world and the COVID applications of nanobodies, and they really highlight the power of this small format. The successful announcement today follows on from a group in the US about a month ago, and a group actually in Australia earlier this year, who demonstrated the ability to bind the spike protein of COVID in ways that the monoclonal antibodies haven't been able to. And they're binding specifically to a conserved part of the spike protein that seems to be consistent from variant to variant. And it's really at the junction of the spike with the virus envelope, which is really a hard spot for a traditional antibody to get to. So we think it really demonstrates the power of the iBody technology and the single domain antibody technology to solve some of these high, difficult drug targeting challenges.
And is it worth just making the point that with AdAlta's technology, whilst you use the scaffold from the shark, which provides those benefits, it's actually a human material, right? So there are benefits there.
That's correct. So one of the pieces of feedback we got very early on in the development of the platform was that pharma would like to see a reduction in the theoretical risk of immunogenicity of a shark-based or a foreign non-human-based scaffold. So our core discovery that underpins the iBodies was the discovery of human proteins that map onto that shark scaffold exactly, but we are using that human protein. So yes, we are the first fully human single domain antibody platform, and we're the first on that shark scaffold to reach the clinic.
And just to plain English it for anybody that's not across the term, so immunogenicity is kind of, we're talking about the body rejecting the... At its
simplest level, yes, correct.
Yeah. Okay, great. Thank you. Okay, so we'll move on to the next question. What does the recent Victorian government R&D cash flow advance mean for you?
The Victorian government has what they're calling an R&D cash flow loan advance program. This is the first of its kind that we're aware of in Australia, and we think it's a fantastic initiative. It's really designed to provide support for technology and research-based enterprises in Victoria that are the backbone of the Victorian economy. It's a low interest loan. It's up to 80% of our forecast R&D spend or R&D tax incentive rebate for FY 2022. It's repayable in 2 years' time, and the interest rate is 0.25% at the moment. So essentially, it's very, very low cost funding that enables us to extend our cash runway in a non-dilutive way and helps us achieve some more of those milestones that I mentioned each quarter than would have otherwise been possible. So we're very grateful to the Victorian government for this facility, and we're one of the first companies to have received the benefit of it.
Thank you. And we've just had a follow-up question on that one, which is how will the Vic government funding arrangement affect the need for equity capital?
Obviously, it's part of our cash flow mix. People will be able to see our 4Cs for the preceding quarters, and we'll see that we generally run at about $1.8 to $2 million average quarterly cash burn rate. So I'll allow people to do their own extrapolations from there.
Okay, great. Can you just talk a bit more about the scope of your collaboration with Carina Biotech and whether it's possible to work with any other companies on CAR-T?
So our collaboration with Carina covers up to 5 targets. That could spin off multiple products because we can combine those different targets in multiple different ways. And as with all of our collaborations, what we tend to do is provide target-specific exclusive or target exclusivity to our collaborator. So that means that we can use the technology or the idea with other targets with other companies. So absolutely, we can engage in other discussions with other targets and other companies in the CAR-T space. And they may or may not be things that we want to work with Carina on as well. So we have a lot of flexibility in that field.
And a follow-on question. Can you just talk to how the IP works around that arrangement?
Yeah, so we have a very clear pathway where we essentially share the costs of getting to what we call proof of concept. And proof of concept means we've designed an Ibody-enabled CAR-T cell that works against the tumour of interest in a mouse. So we share the costs up to that point, and we share the IP that results. From then on, we can either out-license to a third party, we can continue to co-develop and share the costs and hence share the IP, or we can license the technology to either party to progress development from there, and we've predetermined what those licensing terms will be.
Great, thank you. Question, similar sort of question, but now on 8214, which is your fibrosis drug, of course. So what would an ideal partner look like for you on 8214, do you think?
So we're looking at a couple of options with 8214, actually. An ideal partner would obviously be working in the fibrosis space already, and preferably with some respiratory capabilities as well, particularly able to exploit the inhalation version of our drug. We're also looking, though, at partnering for the Asian region separately to a global partnership, and that's important because we can potentially do that a little bit earlier than we could with the global deal. And we think we could get a local partner who's probably going to have a better capability in that particular region than necessarily a global partner who's based in North America or Europe. In an ideal world, the partner would also have interest in other indications that would also then enable us to fully realise the potential of 8214. But with the early development of an inhaled route of administration, we're also thinking really, really hard about partnering by indication. So 214 for IPF could well then become a partner for respiratory disease, and then we pick someone else for example, eye fibrosis down the track with a completely different formulation that can't be interchanged in the marketplace.
Great. Okay, we're just about out of time, so we'll make this one the last question. Given the respiratory nature of 8214, do you think that it would have application in this condition that's been called LongCovered?
We don't know. The short answer is we don't know. What we do know is that patients who suffer from acute COVID or similar SARS, MERS type infection do end up with fibrosis in a fairly significant proportion. What we don't know yet is whether that's going to evolve in the same way that IPF does, in the sense that it's continuously progressive and degenerative, or whether it does recover. But certainly the idea that long COVID is out there, it's becoming increasingly apparent that that is of growing concern, would suggest that there are going to be long-term health consequences. There's certainly been a lot of suggestions early on in the COVID pandemic that we needed better antifibrotics, both for managing COVID infection in the acute phase, but also for dealing with these long-term fibrotic effects. And we do know that Boehringer Ingelheim and other companies who are some of the leaders in the space today are actually starting those studies to explore what happens. So we're monitoring those very closely, and that would be a logical indication extension for us. Similarly, cycling back to the early conversation around antibodies, we made a strategic decision back in 2020 that we were not going to pivot to becoming a COVID company, because we simply weren't ready and able at that point, and we would have been jeopardizing our core programs around AD214. That's not to say we haven't been monitoring that, and it's not to say that there may not be opportunities down the track to look at other ways of focusing on COVID. But for us, it's really something that says we want to do that in a smart, sensible way that does not detract from our core strategy.
Makes sense, Tim. That's great. Okay, well, listen, we might thank you at this stage for both your time and all the thought and effort that you actually put into responding to the brief for today's event. So sincere thanks for coming along, and I'm sure there are lots of people here who will keep an eye on what you're doing. So thank you.
Thank you very much.